Um, this title was chosen really because that's really how we thought about the problem, and you know, physicists, especially physicists working in nonlinear optics, can always invent all kinds of very clever techniques that maybe can make a very nice physics publication, but is unlikely to ever go into a clinic or a hospital. And so we kind of thought, well, what's the simplest possible thing we can do? And so that's what I'm going to try and tell you about today. So I don't think I need to say anything here about nonlinear optical microscopy and all the advantages that it has in terms of spatial resolution and being inherently confocal and so on. You've met plenty of car salesmen, I'm sure, in this seminar series. This is just meant to sort of summarize what people argue are the advantages of doing cars microscopy, and the argument is that, well, it's a label-free nonlinear optical microscopy that has chemical sensitivity, which obtains from the fact that you use a Raman resonance, and Raman spectroscopy is sensitive to molecular vibrations, which fingerprint the molecule. And so that's the argument that people make for this application. And there's some reason to do so. However, difficulty is conserved in life, and so whenever you think you have a good idea, there's an equally bad idea that seems to make your life difficult. And in this case, one of the largest difficulties, aside from the usual difficulties that one has to consider, like photo damage and so on, which are always an issue in any laser-based microscopy, the issue is what's called a non-resonant background. And so we have this diagram, which I'm sure you've seen many times before, where I send in two colors of light, this yellow and orange one, and I require their frequency difference to match some mode, some Raman resonance in the medium. And so the rest of this is just sort of generating the signal, which is this blue color which comes out, and that's what we measure. However, I have all these colors there at the same time, and so I cannot prevent this from also happening. And this is, again, this is two up photons and one down. There's two up and one down. I've just changed the time ordering of these things, so I actually cannot prevent this from happening. The only difference is that this has a resonance and this doesn't. So that's called resonant and that's non-resonant. But there could be only a few of the resonant molecules in your focal volume, and it could be plenty of these ones. So these ones can even dominate. And the trouble is that they both emit the same color of blue light, and they interfere with each other, and that's sort of a headache. So how do you deal with this? Well, the obvious argument you could make is, well, this non-resonant background reduces the contrast, and it's not molecule specific. So what you'd like to do, therefore, to improve the contrast is at least you want to make sure that all your photons are on resonance. And so this was the argument made about 10 years ago by the group at Harvard, saying that, well, what you want to do is you want to make sure that all your photons are on resonance, and so therefore you want to use a picosecond laser which has a narrow bandwidth so that all of your colors are on resonance. And femtosecond pulses, which have 10 or 100 times the bandwidth, are going to be much too broad, and this is not going to work. So the argument goes like this. They looked in the fingerprint region. This was a polystyrene bead at 1600 wave numbers, CC fingerprint region, and you have a line width of 5 to 10 wave numbers. If you were to look in the Raman spectrum, you'd see something like that. So the argument was, well, if you come with a picosecond pulse whose bandwidth matches that, you're going to do a much better job in terms of getting the resonant response. Whereas if you come with a femtosecond pulse, well, it's just too broad, and this is going to be terrible. And so for a decade or so, this was sort of the argument that was made, that you simply shouldn't do cars microscopy with femtosecond pulses. However, the first point I'd like to make is, well, these Raman lines have line width that sort of depends what you're looking at. So for example, if you look at a lipid stretch here, it's around 100 wave numbers broad. If you're interested in biological water, that could be 400 wave numbers wide. So it's not necessarily true that a picosecond pulse is the optimum line width. So what is the best laser line width to choose for cars microscopy? Well, I think the answer is it depends. It depends what you want to look at. So this is a simple calculation where we are looking at the resonant versus non-resonant response as a function of the line width of a transform limited laser. And what you see is that where the dark line crosses the green line depends on what mode you're looking at. So, for example, if you're looking narrow line width, then basically, if you want to be above this green line, which you would like to do, so that your resonant signal is the dominant one, then you have to think a little bit. So there's another way to say this, which is that what is the efficiency of the signal that you want to generate? So obviously, if you have a very high contrast but zero signal, that's not very useful. Equally well, if you have a very high signal with zero contrast, that's not very useful either. So you want the best of both. So we define this sort of efficiency as simply the contrast times the signal, which we are going to take as the resonant divided by the non-resonant. That's the contrast. But we want some signal, so we're going to multiply it again by the resonant signal. So I'm going to replot what I did before and show you something which is sort of intuitively obvious, which is that you get the best efficiency for any given mode when the bandwidth of your laser matches the line width of that mode in the material. It seems sort of obvious, but this is just sort of a demonstration that in other words, if you have a CC stretch, you would be best using a 3 picosecond pulse for your microscopy. However, if you want to look at a CH stretch, you might want something that's like 120 femtoseconds or something, and if you want to look at the OH, actually you're better off with a 40 femtosecond pulse, just based on this efficiency argument of matching the line width of your laser to the line width of the mode you're interested in. However, there's other things that you'd like to do. I mean, this is non-optical microscopy, so you have other kinds of signals you can generate, like second harmonic and third harmonic and various other things. So some of those are non-resonant, like second harmonic generation is non-resonant. 2-photon fluorescence could be electronically resonant. But these things basically benefit from much shorter pulses. You have much brighter images with a femtosecond pulse than with a picosecond pulse. So depending on the problem you're looking at, you might like to have some trade-off between these non-resonant microscopies like second harmonic generation and cars. And that trade-off you might like to be able to adjust yourself depending on the sample and what you want to look at. If second harmonic is really important to you, you might want to be able to see it in this image. You don't want to have to then bring it to another laser and another microscope to scan it again. So, in other words, what you'd like to have ideally is to have some sort of adjustable pulse duration so that you, the user, can optimize this contrast and resolution based on the sample and the problem that you have at hand. So that's the question we had, is can you do this? And how would you go about doing this? Because to have a laser that is adjustable from femtoseconds to picoseconds, sort of under user control, ideally under computer control, is not so easy to do because picosecond and femtosecond lasers are literally constructed very differently. So we have what I call an embarrassingly simple approach, which allows us to do sort of all these things. But basically it allows us to do many of the things that can be done with this sort of standard picosecond sources that people have had. But it's not based on picosecond pulses. It's a really very simple one laser approach. In fact, as I'll show you, we've even done this now with fiber lasers, and so we think that this is something that can really be packaged with almost telecom sort of stability. And as Eric mentioned, it's now a commercially available product from Olympus Corporation. Okay, so this is our Cars Made Simple paper, and we called it Optimally Chirped. You know, it's one of these physics titles that you choose, but I'll explain what this means. It's actually a very simple idea, and not a new idea. It's an established, very old idea in nonlinear optics. So this is a cartoon of a 2-photon microscope, let's say second harmonic generation, and what you would do is you have a femtosecond laser and you raster scan across the sample and you collect, in this case, the second harmonic light. To do cars, we need 2 colors. So we need to generate this second color. And what we don't want to do is have to buy another laser and synchronize it and do all that sort of stuff. That's expensive and probably requires somebody with some capability in optics. You want to do the simplest possible thing. So how can we do this Stokes generation that is required for the technique in the simplest possible way? And so we think that one of the simplest things you can possibly do is take advantage of developments in nonlinear optical fibers, photonic crystal fibers. And so these things are, they're also known as holy fibers. And this has different meanings for different people depending where you come from. But nevertheless, the design of this allows you to control both the nonlinearity and the dispersion simultaneously. So you can engineer these things. And also, because it's a single spatial mode, it removes all kinds of space-time coupling effects that happen during nonlinear optical propagation. So I'm just going to make a few brief comments. There's a very nice review article from 2006 if anyone really wants to read about this stuff. But basically there are two parameters, two length parameters in this nonlinear propagation. One is called the dispersion length, and the other is called the nonlinear length. And what happens is you get soliton formation. And if you have too much nonlinearity, you can have multiple solitons, and these solitons split. It's called soliton fission. And these can interfere with each other in horribly chaotic ways. And what you get out are essentially extremely noisy spectra. And this problem is sort of characterized by the soliton number, which is related to the ratio of those two things. So to make a long story short, what you'd like to have is to minimize soliton fission so you get very stable spectra. And that means keeping this small. This number is small, which means you want to maximize this and minimize that. No. You want to minimize the dispersive length and maximize that. You want this ratio to be as small as possible. I hope I said that right. Okay, so let's think. People dismiss this because people that tried this in the past said this is terrible for microscopy. It's very noisy and doesn't work at all. And so basically they threw out the baby with the bathwater. But this is actually really well understood physics, and it really depends, as I'll show you, with great detail on how you set it up. So if you have a photonic crystal fiber with one zero dispersion point, and if you pump on the blue side of that zero dispersion point, you can get some pulse broadening. In the usual way, you get self-phase modulation. But the spectral broadening is very limited, and the broadening is not sufficient to really generate the second color that you need for cars. For example, you could never generate the bandwidth you need to look at CH or OH or something like that. So you don't get soliton formation in this case, and you get a very stable output because it's just all SPM. But it's not useful for our application. If you pump, however, on the red side of that zero dispersion point, then you have an anomalous group velocity dispersion, and you get all kinds of things happen. You get Fourier mixing, you get self-phase modulation, you get soliton formation. You do get a red-shifted continuum, and that's what you want for cars. But you have these high soliton numbers, soliton fission, and you get what's called modulation stability, which is basically the amplification of fluctuations. And it's unavoidable. You can't avoid this thing. And you get a horribly noisy spectrum. This is just a disaster. You just don't want to do this. And this is why people say PCFs are noisy, it's because of this modulation instability. Now, if you have a PCF which you've engineered to have two zero dispersion points, and you pump in between them, then what you get is you have a soliton propagation, but then the soliton sort of, as it's redshifting, runs into the second zero dispersion point, and what it does is it couples out, it sort of spills out the side, and you get a redshifted dispersive wave. And that redshifted dispersive wave, which is just on the red side of the second dispersion point, which you can engineer to put it where you like, is good for cars, and you can have a very stable output because the soliton propagation length is small. Basically, it just runs into that second zero dispersion point and couples out. It's no longer a soliton. So this can be very stable output. So let me summarize. With these things in mind, picosecond versus femtosecond pulses. With a femtosecond pulse, you have a much broader bandwidth, and so the dispersion length is very short because the pulse basically spreads out really quickly because of dispersion. And this means that it's easy to keep the soliton numbers small, and you can have very good stability. With picosecond pulses, because of their narrow bandwidth, they propagate much further without dispersing, and you have a larger soliton number propagation. It's very, very hard to get good stability of the output with picosecond pulses. And again, if you want to read about it, you can read there. So we have chosen this particular fiber, which is actually commercially available, and it has these two zero dispersion points, and these are convenient because we're going to pump at 800 nanometers, so we'll be in between the two, and we'll get this red-shifted dispersive wave on the red side of that, which is very nicely convenient for lipid imaging, water imaging, and so on, because it's going to be around 1,000 nanometers, which is sort of a 2,500 to 4,000 wave number shift. So that's exactly what we want. So there's the output. And if you start with 800 and you want to look at lipids, 2850 wave numbers, then that means that the second color has to be 1050 nanometers, and that works quite well, and we have some tunability. And in the anti-Stokes generation process, you go as much to the blue as you did to the red, and this would make the signal that we measure at 650 nanometers, that's a very convenient signal to detect with a PMT. And so this looks reasonably promising. However, there's an issue here which comes back to something we brought up earlier, which has to do with line width and resolution. I talked about femtosecond pulses being broad, and that wasn't good for cosmic microscopy. And what I did is I generated this femtosecond pulse here, which is way broader. So this, I may be able to generate this stably, but the line width looks totally unusable for cosmic microscopy. This looks like a disaster. So how are we going to deal with that? Well, before I get onto that, let me just do one more thing, which is I'll show you the effect of the pulse duration on the noise spectra. So we're going to measure the noise in these photonic crystal fibers with this sort of setup. So what we have is a femtosecond laser, which we can control the chirp, and we send it through a photonic crystal fiber, and then we send it into a photodiode, which we measure with an electronic spectrum analyzer, and we're going to look at the spectral noise density in the RF, the noise in the measurement, as a function of the band that I look at. I'm going to resolve within this continuum, and I want to see how it depends on the input properties of the chirp and the pulse duration. Okay, so it's wavelength resolved noise measurements, and we're going to look with a spectrum analyzer over this 9 kilohertz to 1.5 gigahertz bandwidth. If you think about this, 9 kilohertz is 33 microseconds. So if you have a dwell time that is less than 33 microseconds, then you're within this band. So those are the noise features that are relevant to microscopy. Okay, so the first thing you can do is just take this on a photodiode, and if you look at the pulse train from the oscillator in black, you see a standard deviation of 1.3%. Not bad for an oscillator. If you take the total continuum from the photonic crystal fiber without wavelength resolving it, it's pretty comparable. You get 1.7%. So you think that doesn't look so bad. But if you actually take a slice, a 10 nanometer slice at 1050, which is what you count, what counts for microscopy, it's terrible. This is this blue here. That's almost 40% shot to shot. That's a disaster for live cell microscopy. So that really doesn't look good. And this was with an uncompressed 220 femtosecond pulse being input into the photonic crystal fiber. And this is what you get. This is one of the reasons why people say, photonic crystal fibers are not good for microscopy. However, I want to show you the following. This is now the measurement of the with the 220 femtosecond pulse. Now, where I'm looking spectral resolved. So the gray line is the noise floor when I block the laser. And this is 9 orders of magnitude down. You can see that even though I thought I blocked the laser, I didn't block it perfectly. I have a little bit of a spike there, 9 orders of magnitude down. So it's a very sensitive measurement. And so now what you can see is if I look at the total continuum, which is black, you can see that it's almost the same as the noise floor from the laser itself. So the spectrally integrated continuum looks very good. But then if I take a slice, and I want to make a comment that this is at constant power, so there's no change in the Poisson noise here. This is at constant power. And what you see is that the noise increases by 3 orders of magnitude when I take a slice. It actually depends on which slice you look at. Okay? So that's the problem for microscopy, is you need to look at the noise in a given narrow bandwidth. So what's interesting, I'm just saying that what I just said. Now what I'm going to do is I'm going to look at the noise density in this 10 nanometer bandwidth around 1050 as a function of the input parameter of the laser. So there's the noise floor, and if I take a transform limited 70 femtosecond pulse, actually, what you see is, before I showed you a 220 femtosecond pulse, this is now 70 femtosecond pulse at the input to the fiber. So you have to control the dispersion such that at the input to the fiber it's short. And then you see that in this band, actually, I'm almost as good as the whole continuum. But if I let that transfer limited 70 femtosecond pulse chirp to 90 femtoseconds, the green one, the noise increases by almost 3 orders of magnitude. And this is the modulation stabilities that are building up. So in other words, if you're not really, really careful with controlling the chirp at the input of the fiber, you can increase the noise by orders of magnitude. If I take that 70 femtosecond pulse and instead make a transfer limited 100 femtosecond pulse, the noise increases by more than an order of magnitude. If I let that 100 femtosecond pulse stretch out to 105 femtoseconds, which doesn't seem like much, you can see the noise increases by almost an order of magnitude. So in other words, it really is sensitive to the input parameters. But if you really keep your pulse short and transfer limited at the input, this keeps the Soliton numbers very low and you get stable output. Okay, so are PCFs noisy? It all depends. But they can be made to work very well. Okay, now the second issue is, how do I deal with these very broad bandwidths? And so what people have tended to do is to spectrally filter them. You say, well, you know, I need to have good spectral resolution, so I'll have to somehow get rid of all this extra bandwidth, which is kind of crazy in a way because you spend all this time trying to make it, and now you're going to throw out all your photons. So it's kind of wasteful, and actually, it turns out it's unnecessary. And the reason why it's unnecessary is because this is nonlinear optics. And the spectral resolution is not just simply related to the line width of the lasers. You actually have to think about the various processes that govern the resonances. And this is a resonance at second order. It's an up and a down process. And for cars microscopy, what we're concerned about is the line width at second order. In other words, the Raman line width. So, in other words, we could take advantage of things that are extremely well known in nonlinear optical spectroscopy, which is that you can control the phase of these light pulses, and the phase really matters. Pulse shaping ideas, okay? So this is sort of a cartoon that if you can control the phase of these different colors, you can make different colors arrive at different times, and there's many ways of doing this. This is called a spatial light modulator, but there's other ways of doing this as well that don't require a spatial light modulator. And the point is that you have a knob you can turn. I can play with the relative phases of these different colors, and this is an important knob in nonlinear optical spectroscopy and in microscopy. So here's the simplest possible idea. The simplest kind of pulse shaping you can do is just called chirping, which is stretching out the pulse. So we, I think, agreed that the Stokes pulse was way too broad, and that was really the problem. There was just way too many colors there, and we weren't going to get good Raman resolution. So I say, well, let's stretch it out in time. You can do this by just linear dispersion, but we're going to keep our pump pulse short. So what the pump pulse does is it doesn't see all those colors, it just sees a sort of gates, this part of it here. So in fact, what limits my resolution now is just the line width of the pump pulse, not the 100 nanometer wide continuum that I have there. So that looks sort of promising, and in addition, it allows for one more thing, which is you can easily do spectroscopy now just by changing the time delay. And just by changing the time delay between them, I can tune that Raman resonance. So that looks kind of good. So we tried this, and what you can see, in fact, is it does work. When you don't do this chirping, this is a drop of methanol, liquid methanol, on the cover slip of our microscope. And so if you don't do this chirping technique, what you see is this broad, this sort of thing, and you clearly have some response, but there's no spectroscopic information there. If you do this chirping technique on the Stokes, then you see, in fact, that you do start to resolve, and these are the well-known, two CH stretches in the methanol Raman spectrum. So that's starting to look kind of promising. But in fact, we can do much better than this. And what we can do much better is actually, if you think about it, what you want to do is you want to chirp both of them. So if you chirp both of them, what are we doing here? Well, we have a Raman resonance which depends on the frequency difference between two colors. So there's color number one, there's color number two, and the Raman resonance here depends on this frequency difference. If I stretch both of them, but I do it a little bit thoughtfully, I pay some attention to how I do this, such that I chirp them at the same rate, then you can see if I change the color on this red side here by some amount delta, and I do it the same for both pulses, the difference frequency is constant. So this pair and this pair have the same difference frequency. And the same is true on the other side. If I go to the other side, if I did it the same rate, this is, again, a constant difference frequency. And that's a requirement for a car's resonance, is you have a constant difference frequency. So now I have all the colors in the pulse are all there at the same time, but the difference frequency is constant. So now, if you want to think of it in the following way, you can also make these frequency time plots, or Wigner plots if you like. And so when I don't have the chirp pulses, I have this broad bandwidth, and they're very short in time. So this is the difference frequency with just simply taking this and subtracting that. And the width here would be my spectral resolution. But if I chirp them, if I stretch them out like this, you can see that I get a very narrow difference frequency. And in terms of doing spectroscopy, here again, if I have a narrow line width, if I chirp it here, I have a narrow line width. And now when I change the time delay, you can see that the distance between these two will get smaller. And this tunes the Raman resonance. So I can not only chirp them to get a high resolution, but I can easily do spectroscopy just by changing the time delay between them. So when you do that, you get something that looks like this. Do you remember that methanol peak I showed you before? Well, now it's completely resolved. And there is a peak of DMSO, so it has a different frequency. The way you can think about this is if you're doing cars microscopy or spectroscopy with picosecond pulses, it's like you're going to play the piano and you're going to play two keys at the same time, and you're going to keep your fingers a certain distance apart. But you're only going to play two keys, because that's the Raman resonance you want. What I'm saying is here is actually we have a musical score. I'm going to play all the keys in the piano, but just two at a time. I'm going to keep my fingers the same distance apart as I'm going across the piano. So I'm going to play all the keys in the piano, but only two at a time, and keep my fingers the same distance apart. So I'm using all the light that I've generated, and I can easily do spectroscopy just by changing the time delay. There's a demonstration. So that actually works. And so this is an example of how the spectral resolution of this drop of methanol depends on the degree of this chirp. So we can do spectroscopy, and we can choose the chirp, which is essentially the effective line width. So this is a demonstration of what I said before, is that by controlling the degree of chirp, I can essentially go from a picosecond to a femtosecond experiment under computer control. I will. So we can choose the resolution that matches the problem. Okay, so what do we have? What we have is essentially a two-photon microscope. You think about it, I have a femtosecond laser, and I have a laser scanning microscope. And I have this photonic crystal fiber, some pulse shaping. I can collect second harmonic cars in the forward direction, the backward direction. I can collect some fluorescence, so it truly is multimodal imaging. But in a sense, this is really simple. This is just a two-photon microscope with some box of optics in between. And that's all it is. So any two-photon microscope can do this. You just need to have a little box of optics. In fact, this is what Olympus did, and that's why they became interested in this, because there's a large base of installed two-photon microscopes, and we showed them that they could just make a little box of optics, and every two-photon microscope can be a Cars microscope without giving up the two-photon capability, because that's there still. So this was actually a product released in October 2009. I heard rumors that they may have actually sold one. The other thing they did is that together with Olympus, they launched a user facility together with the NRC, and if you want to hear more about that, you can look up carslab.ca. Okay, so this is some snapshots of different things that we can do. I'm not a biologist, but this is a couple of the collaborations that we've had, and this is an atherosclerotic rabbit aorta. And what you see is, this is a dead scan, so we're scanning through the sample about 50 microns deep. And what you see is cars, which is red, which is the lipid droplets. Blue is the collagen, second harmonic. And green is the elastin, which is two-photon fluorescence. And this is sort of a 2 frame per second imaging. And so we can very nicely sort of resolve these things. And our collaborators are quite happy, it seems, with this kind of imaging. The other thing you can do is you can look at myelin. There's a lot of people that are interested in myelin imaging. This is a particular application towards MS. There is no marker in the blood for MS, so people wonder if MS is actually maybe a physical degradation of the myelin rather than an actual chemical modification. And so we have a project in that. This is just, again, demonstrating label-free imaging of myelin. Myelin gives very nice signals. It's an ordered lipid and gives great images in Cars microscopy. This is lipid trafficking in hepatocytes. I have a colleague who's interested in hepatitis C virus infections. And this is just showing how you can track in a label-free manner the lipid droplets along a tube limb. The green is the two-photon fluorescence, the red is the cars. So you can make movies over time of lipid trafficking. And here's another example. This is just purely cars now, and these are live human hepatocytes. So we do experiments where we keep these things alive for 48 hours. We transfect them with a hepatitis C virus replicon, and we look at how the viral RNA sort of progresses and develops inside the cell. We actually have some interesting ideas about how this works and why hepatitis is actually a disease of the liver. So anyway, you can have fun watching these cells move lipids around. Okay, this is something that relates to some of the work done here in our Potman's group, which is that you don't have to look at living things. You can also look at other kinds of biomaterials. In fact, you can look at inorganic samples as well. Whenever you're interested in the spatial distribution of organic materials and you want to have chemical sensitivity, this technique works quite well. So this is more of a biomaterials application. So we're going to look at starch, cellulose, and we're going to actually do some cooking since it's lunchtime and it's quite appropriate. I just mentioned passing that there was a very nice paper that came out before we did our work, actually, by the Potman group. And I encourage you to have a look at that. Okay, so these are potato starch grains, and we're doing simultaneous cars and second harmonic. And again, just to show you our spectral resolution, there's a drop of DMSO, and there's the potato starch grain, and there's the carbohydrate OH stretch, which we're going to be looking at. And these kind of striations are very well known. These images in the second harmonic are known that the sort of crystallinity, the ordering in the starch, sort of varies spatially. These things are well known. People have seen this before. So we decided we were going to cook some potatoes. So what we did is we took these starch grains and we put them on a cover slip and we sealed it and we heated them up to 80 degrees Celsius and we made a movie. And what we're doing is we're looking at the second harmonic in green and the cars in red as we're heating this up. And so what we're watching is how hot water is attacking these starch grains. We can also do spectroscopy here, and you can see how not only is the morphology changing, but the spectra are also changing during this process. And so this is a rice starch grain. So this is actually kind of a pretty picture. This, for those of you who care, this is sushi-grade rice. So what you can see here is that the second harmonic image shows that the crystallinity, the ordering disappears first, and then the swelling takes place afterwards. You sort of watch the two images side by side. You can see that the second harmonic image disappears more quickly than the cars. And it sort of makes sense that you would lose order before the thing swells. And another thing we learned is actually the water actually attacks from the inside, which is kind of interesting. I'm not sure if anyone knew this. But if you're in food sciences, you might be interested in knowing these kinds of things. So we can make movies of cooking some potatoes and rice. Something that relates more to the work from the Potman group is, again, looking at these cotton fibers. And we also found, as was published by Eric and his group, that the orientation of the fiber really matters. And what we think is that the OH band also shows a significant sensitivity to the fiber orientation. So if you take a fiber that's got a kink in it and you look in these different regions, you can see that the ratio of the CH to OH stretch really changes depending on where you look. So there could be some interesting applications in biomaterials for this kind of spectroscopy and microscopy. Okay, so I'd now like to show you that with this cars made simple approach, there are other things you can do with this once you start to think about it. I'm just going to show you a few illustrations of other things that we sort of tried to do since this is working in our lab. We said, what else can we do with this idea? So I'm going to show you that you can do frequency modulation cars, but you can do it in a new way, which is you can do rapid spectral scanning. You can do spectroscopy using FM detection. You can do multiplex cars, which means measuring the entire Raman spectrum, but without dispersing anything, just using a single element detector. If you do photon counting, which I'll show you, then you can do lifetime imaging cars. And finally, if you replace the femtosecond oscillator with a fiber laser, you can actually do an all-fiber cars. So I'll just show you these things. The first thing is the frequency modulation cars. The idea is that it was first developed here by the Harvard group. Is essentially you are sort of hopping on and off resonance at a high frequency, and you're sort of taking a difference image. And this is the idea is to get rid of this non-resonant background. So in our case, spectroscopy is obtained just by time delay, right? The way I do spectroscopy is I change the time delay between the pump and the Stokes, and we put it on a stepper motor under computer control. We literally move this motor. But if instead of doing that, I just put this this pulse on a shaker and I oscillate it, let's say to kilohertz, then what I'm getting actually is a true frequency sweep. Right? So that's a true FM measurement. And so then if we lock in at the frequency modulation, then that's FM detection. It has all the advantages of FM that you're familiar with. That's why FM radio sounds better than AM radio. So this is how we do it. For those of you interested in optics, is we can, as always, we control our pulse duration at the input to the PCF, and what we do is we then split the pump pulse into two, and with 2 time delays, and this is what allows us to do the FM. We're going to hop back and forth between these 2 time delays, Raman shift number 1, which is this resonance, and Raman shift number 2, which is the background, and this one I can scan while I'm doing this. So I can hop back and forth at 5 megahertz, and I can change the time delay here either slowly or at, let's say, 1 kilohertz. And so, again, use sort of the benefits of FM detection. These are fluorescent polystyrene beads in a fluorescent dye solution. And so the contrast is not very good. The signal to noise is maybe 1-to-1. But when you turn on the FM, you really can improve the contrast by at least an order, maybe a couple orders of magnitude. And we sort of think, actually, that this should be a shot noise limited detection scheme. And the main point is that we can do spectroscopy. We can tune while we're doing FM detection. So it's just an example of that. Something that we actually thought about, which I'm pretty sure works, but we didn't actually try it in the lab yet, but I'm pretty sure this is right, is that what we're doing is we're doing this frequency modulation, and we're using a lock-in, and we're detecting a certain phase of the lock-in, and that gives us a certain Raman resonance. But if you think about it, as this mirror is oscillating, you're sweeping through all Raman resonances, right? You're sweeping across the entire Raman spectrum. And so instead of using a lock-in detector, what you should do is just feed that entire thing into an A to D card and digitize the whole thing rapidly. So what you have then is a signal which just follows the entire waveform, but that contains the entire Raman spectrum. And so what you can do is you can get the entire Raman spectrum all at once, but you're only using a single element detector. You don't have to disperse anything. You don't need to read out a CCD camera. So in fact, it's very easy to do multiplex cards in this way. In our setup, particularly because we are doing lifestyle imaging, what we found is these hepatocytes are very sensitive to the laser light. We really had to turn the laser light down before we convinced ourselves that the laser was not modifying the experiment. We quickly realized that our photons are basically coming one at a time. Our anti-Stokes photons are coming at less than 1 per laser pulse. And that was perfectly fine for imaging because the laser is at 80 megahertz, and that was plenty of signal for us. But if you realize that the photons are coming one at a time, then you're making a great mistake using analog electronics to detect these things, because you're just adding all the analog noise into your measurement. For example, if you have a PMT, you're adding the pulse height distribution of the PMT into your measurement. And you're not interested in that. There's nothing that you want to know about the pulse height distribution. It just adds noise. However, if you count the photons, then you completely remove the pulse height distribution, and you now have a digital signal which removes all this analog noise, and in fact significantly improves the signal to noise in the measurement. However, if you think about it, once you are counting photons, then you may as well get more information out of it, and so you can very easily implement time-correlated photon counting. And so this is what we did, and it also actually gives you another contrast mechanism, because on the electronic time scale, CARS is instantaneous, whereas fluorescence always has a lifetime. So anything that arrives after time 0 in your instrument response function cannot be a CARS signal. So it gives you another sort of almost for free contrast mechanism for CARS microscopy. So I'm going to show you some images where we look at these liver cells again. We're going to use CARS to detect the lipid droplets, and we're going to use two well-known stains. I'm going to show you how we can simultaneously do FLIM, fluorescence lifetime imaging, and CARS. Again, all with this very simple setup. What we're going to do a little bit differently, though, however, is we're going to do forward-collected CARS and FLIM, because the fluorescence goes everywhere, but cars really is forward. And so we have a problem, which is that we want to collect the non-descanned detection. And if any of you are familiar with time-correlated photon counting, these detectors are tiny, right? That's how you get the good time resolution. And so when you're scanning, you're going to be sweeping on and off the detector. And so that's why people have done all kinds of things like try and collect in the backward direction by having a mirror and so on. It all gets really complicated. But it turns out that you don't have to do that if you, again, just think about it just a little bit, that you can collect the non-descanned forward detection. You can do it in the following way. So here is the microscope. There's the objective lens. And so I have this raster scanning here. What I'm doing is I'm changing the angle such that at the focus of the objective, I have a different position. And that's how I do my scanning. Now I have the condenser. And it turns out that there's a conjugate plane right here where you have another focus. It's another focus where, again, you have all these come to one point in space, but at different angles. So what you want to do then is you want to image that plane into a multimode fiber. And you want to choose the multimode fiber so that its numerical aperture matches the range of input K vectors that you get from here. And if you do that, then all the light that is being raster scanned does couple into the fiber, because the position is not changing at this plane here, but the angle of incident, the angle is changing. And as long as the numerical aperture of the fiber can collect all those angles, then it should work. And so that's the idea. So we used a 600 micron core with about a 0.4 Na. And for those of you who are familiar with time-correlated photon counting, what you might worry is, because everyone will tell you can't do this with multimode fibers because of modal dispersion, right? You're going to spoil your time resolution. And what I'd like to show you is that that's actually not true. So this is the non-descanned forward collection. And what I can show you is this is with a PMT. This is not time resolved now. This is just collecting the total light. And this is the image you would get of this pollen in water. When you just put a PMT, you collect all the forward non-descanned light. And this is what you get when you put the multimode fiber at the conjugate plane. And you can see it's not perfectly the same, maybe a little bit of a loss there, but it's really not too bad. Right? This is several hundred micron by 100 micron field of view. So the field of view is not really a big problem. So then the next question is, what about your time resolution? Have you spoiled your time resolution by having this multimode fiber? So this is a car signal from a drop of oil. And what we do is we get this bright spot, again, hundreds of microns across. And what we're going to do is we're going to look at the instrument response function as a function of how far we are off axis. Because the idea is that as you go further off axis, you can imagine that you're launching higher order modes in the multimode fiber. And what we can see is, yes, there is an effect. We can measure it. We do see some modal dispersion. But the dispersion is about 17 picoseconds over this field of view. And so the spread, in other words, of the instrument response function is about 17 picoseconds over this range of view. And it's maybe not perfect. If you're really worried about it, maybe you wouldn't like that. But for our purposes, that's good enough. So now what that allows us to do in a very simple way is to combine cars with lifetime imaging. And so here's an example. We're collecting the total image in the forward direction, non-descanned, and we're binning them now just according to lifetime. So anything that's instantaneous is cars, and so we see that. And then if we then look at with these lifetimes here, we can see two different dyes, and we can sort them out. And the reason why we're interested in this is because we are working on colocalization problems where we want to look at a cars image and a fluorescence image, and you know, we would like to draw some mechanistic conclusions as to what it means when these images are colocalized. The trouble is that in cars, our spatial resolution is, let's say, 300 nanometers or so at best. And if I have some molecules fluorescing and some other molecule that's giving me a car signal, and they're 100 nanometers apart, I would say they're colocalized based on my cars microscopy, or this correlation microscopy. But chemically, they don't know anything about each other. They're 100 nanometers apart. That's a universal way. So I would mechanistically either draw the wrong conclusion, perhaps, or not be able to make any conclusions. However, if I can combine that with a lifetime imaging measurement, and I know that for something that affects the lifetime, I really have to have, like in FRET, if you're familiar with FRET techniques, I really have to have a chemical interaction, something within 5 nanometers that's going to tell me that there really is some bonding event or some Stark effect, if you like, from some countercharges on a protein, which is modifying the fluorescence lifetime. Then I can use this technique as sort of like a coarse and a fine grain, like a vernier. I can say that with cars and two-photon fluorescence, I can say that they're within 300 nanometers of each other. And if in addition I see that there's an alteration of the fluorescent lifetime, that I can say that not only they're within 300 nanometers, but they're also within 5 nanometers of each other. And so I think this will help to sort of develop mechanistic models of what's actually going on. Okay, last thing I want to tell you is that, we like lasers, and lasers are fun, but let's admit that these kind of lasers are not yet ready for going into hospitals and clinics. And eventually all this stuff is going to be done all with fiber lasers with telecom stability, and that's the way it should be. And so we actually had a collaboration with a laser company, fiber laser company called IMRA, and we did a demonstration of this approach to all-fiber carbon microscopy, again using a photonic crystal fiber. In this case, it's a different kind of fiber. It's something called a suspended core fiber. So it's not the holy fiber that I showed you before. It's got this kind of geometry and has a much higher nonlinearity. And this allows us to get the Stokes generation at much lower powers because these fiber lasers are lower power. So this is a second harmonic of an erbium doped fiber. We pumped this ultra-high nonlinear fiber. You can tell that it was some marketing guy who came up with this name. Ultra-high nonlinear fiber, anyway. But it's the same idea. We get pump and Stokes, and we're going to do our chirp pulse microscopy as we've done before. And it works. So this is the rabbit aorta tissue. This is looking at stained hooked and cars in some liver cells. And finally, we can make movies of lipid trafficking in hepatocytes. Now, what I want to point out to you is we were quite disappointed at first with the noise, because this doesn't look as good as the imaging that we saw with our femtosecond laser. And then what we did is we looked at the spectral distribution noise. We saw the giant spike at 47 kilohertz that was really spoiling our imaging. And so we called up the company and said, "What have you got at 47 kilohertz?" And they said, "Uh-oh." And it turns out it was their switching power supply. So they actually could fix that right away almost, but unfortunately not in time for a publication. But nevertheless, I'm just saying that if you remove that 47 kilohertz noise, this actually is a pretty good imaging system. So I'm fully confident that cosmic microscopy and other kinds of Raman microscopies are going to be done all with fiber lasers in the future. One last thing I'll mention before closing is that there are other opportunities if you think further about what you're doing with pulse shaping, is that certainly if you do pulse shaping in a conventional manner using what are called SLM, spatial light modulators, there's unavoidably a space-time coupling that occurs, and we wrote a paper just basically showing that you unavoidably have a space-time coupling in these spatial light modulators. And for people that are doing coherent control, this is actually a headache, because what it means is that as you are shaping the electric field of your laser pulse, you're also changing its intensity distribution at the laser focus. And if you have some nonlinear signal, which also depends on intensity, then you're doing two things at once, and it's hard to sort them out. And that's a serious question. However, if you think about it from the point of view of microscopy, what you actually are doing is you have simultaneous control over the nonlinear point spread function and the nonlinear power spectrum. And there could be some new opportunities to emerge from having simultaneous control over these two things. And with that, I would like to close and thank my coworkers at the NRC, at Olympus, IMRA, in Taiwan, and in Queen's University, and the chance to talk with you today. Thanks.